Hello students. In our previous sessions, we have learned types of reproduction, structure of flower, development of male and female gametophyte, then types of pollination and pollinating agents. Similarly, we have studied contrivances for cross-pollination, then outbreeding devices. Then we have also studied the process of double fertilization, then post-fertilization event, that is development of embryo and endosperm. In this session, we are going to study the development of seed and fruits, then significance of seeds and fruits. Similarly, we will also study Apomixis and Parthenocarpy. Now we will study the development of seed and fruits. It is one of the post-fertilization event in angiosperm in which ovule develops into seed and ovary develop into fruits. The integuments of the ovule form the covering of the seed called as seed coat. The outer integument forms testa of the seed coat. Testa is thick and hard. Then inner integument forms thin and delicate layer called as tegmen. Tegmen is generally thin and delicate. In seeds of some plants, nucellus remains in the form of a layer called as perisperm. Perisperm is found in plants like beet and black pepper. The seeds of some plants show presence of one cotyledon, that is monocot, or sometimes it shows presence of two cotyledons, that is called as dicot. These cotyledons store food material. The micropyle in the seed coat provides passage to the radicle to come out during the process of seed germination, as well as micropyle allows the entry of water during seed germination. Now we will study the use of endosperm. You know that endosperm is used for the developing embryo after the process of fertilization. Depending upon the presence and absence of endosperm, seeds are of two types. The first is endospermic, or it is also called as albuminous seed, and the second one is non-endospermic seed, or it is also called as ex-albuminous seed. First, we will study endospermic seed, that is albuminous seed. Example, castor and maize. The given diagram shows vertical section of maize grain. We see on outer side the fruit wall, that is pericarp, and seed coat is fused with each other to form a common structure. Inside the seed coat, a layer of protein is present. It is called as alveolar layer. The developing embryo uses only some part of endosperm for its development, and major portion of the endosperm remains inside the mature seed. Hence the seed is called as endospermic seed. The seeds show presence of single cotyledon. It is called as scutellum. Then at the lower side of the scutellum, embryo is present. Embryo shows presence of plumule, which is protected by a tissue called as coleptile. Then lower to the plumule, a radicle is present, and radicle is protected by a tissue called as coleryza. You all know that after germination, embryo develops into a new plant. During the development, the plumule develops into shoot or leaves, while radicle develops into roots. So in this way, endospermic seed is found in castor and maize. Now we will study the second type of seed, that is non-endospermic seed, or it is also called as X-albuminous seed. The common examples are bean and pea. In the diagram we see the structure of an opened bean seed. You all know that bean seed is a dicotyledonous seed. That is, it shows presence of two cotyledons. In bean seed, the developing embryo utilizes the total endosperm for its development. Hence, after the maturity of seed, endosperm is absent. So, the seed is called as non-endospermic seed. The cotyledons store protein. At the top of the seed, we see embryo showing epicotyl, hypocotyl, and radicle. The upper part of the embryo is called as epicotyl. Epicotyl shows presence of plumule. This plumule later on develop into a plant or shoot. The middle part is called as hypocotyl, and the part which is at the lower side of the hypocotyl is called as radicle. You know that after the seed germination, radicle develops into a root. Then for the protection of seed, a protective covering is present. This protective covering is called as seed coat. So bean and pea are the types of non-endospermic seed. Now we will study the development of fruits. After fertilization, the developing seeds produce a hormone which help in the development of fruits. The ovary begins to differentiate into fruit, and ovary wall develops into a layer called as pericarp. The diagram shows typical fruit of mango. In mango, pericarp is differentiated into three layers. The outermost layer, which forms the skin of the fruit, is called as epicarp or exocarp. Inner to the epicarp or exocarp, fleshy, juicy, and edible layer called as mesocarp is present. The innermost hard and stony layer is called as endocarp. As endocarp is hard, it protects the seed. So, fruit develops simultaneously when the seed develops. Now we will study the significance of seeds and fruits. You all know that after the process of fertilization, ovule develops into seed and ovary develop into fruit. You all know that fruits provide nourishment to the developing seeds. Similarly, the fruits protect the seeds in immature or young condition. The seeds are important propagation organs of the plants. That means we can use seed to grow or to develop new plants. The fruits and seeds are important sources of food for animals as well as humans. The seeds and fruits develop special devices for their dispersal and help in the distribution of species to different places. Dispersal means the seeds are transferred from one place to another place by the help of different transporting agents like wind, birds, or animals. So, seeds and fruits play an important role in spreading or distribution of a particular species. Now we will study the next topic, that is seed dormancy. It is the metabolic arrest of the growth of the embryo. It means the growth of an embryo is temporarily stopped or suspended during unfavorable condition. This phenomenon is called as seed dormancy. It is the structural or physiological adaptive mechanism for the survival of a particular species. During the process of seed dormancy, mature and viable seeds are dispersed. Viable seed means the seeds are having the capacity to undergo the process of germination. The seeds are dispersed to different places. It means that the seeds are transferred to different places with the help of wind, birds, or animals. The germination of seeds occur only after the completion of dormancy period. There are different phenomena related to seeds. The first one is apomixis. Apomixis means the embryo is produced inside the seed by asexual method or without undergoing the process of fertilization. That is, there is no union or fusion of male gamete with female gamete. The second one is apogamy. In apogamy, when the gametophyte organ or one of the cell of gametophyte directly develops into embryo without undergoing the process of fertilization, such phenomenon is called as apogamy. The third one is apospory. In apospory, a diploid sporophytic cell which produce embryo without fertilization. You know that apomixis means formation of embryo without fertilization. That is, the embryo is produced without the fusion of male gamete with female gamete. Apomixis is of three categories. The first one is recurrent apomixis. Recurrent means it occurs repeatedly. In this, embryo sac develops from an archisporial cell or one of the cell of a nucellus. In diplospory, the embryo sac develops from a diploid megaspore mother cell. Hence it is called as diplospory. In apospory, one of the cell of the nucellus develops into an embryo. The second category is non-recurrent apomixis. Non-recurrent means it does not repeat. In this type, embryo develops from an egg directly or haploid cell of a gametophyte. This phenomenon is also called as parthenogenesis and it occurs through apogamy. Plants produced by this method are sterile and do not reproduce sexually. Example of it is Nicotiana. Sterile plant means they do not produce male and female gamete. As they do not produce male and female gamete, the process of fertilization does not occur and hence they do not reproduce sexually. The third category is adventive embryony. In this type, embryos may develop from the cells of nucellus or integuments along with normal zygotic embryo. Zygotic embryo means when male and female gamete fuse or unite with each other, they produce diploid zygote. This diploid zygote undergoes repeated mitotic divisions and produce an embryo. Such embryo is called as zygotic embryo. So in seeds or in some seeds, along with this zygotic embryo, other embryos may develop from the cells of nucellus or integument. So, as more than one embryos are produced inside the seed, this condition is called as polyembryony. Polyembryony is seen in plants like lemon and orange. Now we will study the next topic, that is Parthenocarpy. The term Parthenocarpy was introduced by a scientist called as Knoll in the year 1902. In this, condition of fruit develop without the process of fertilization and the seeds are not developed. That means the fruit show absence of seeds. Hence, the fruits are called as Parthenocarpous fruits and the phenomenon is called as Parthenocarpy. The common examples of Parthenocarpy are pineapple and banana. In these plants, the placenta of the ovary produce growth hormone called as auxin. The common auxin is IAA, that is indole 3 acetic acid, is responsible for the enlargement or development of ovary into a fruit, and due to this auxin seedless fruits are produced. Then we will see polyembryony. Polyembryony means development of more than one embryo in the seed. Polyembryony was first noticed by a scientist called as Leeuwenhoek in the year 1719 in the seeds of citrus. Polyembryony is of two types. The first one is called as adventive embryony and the second one is called as cleavage embryony. In adventive polyembryony, an embryo develops directly from the diploid cell of nucellus or integuments in addition to the zygotic embryo. The second type of polyembryony is called as cleavage polyembryony. In this type, the zygotic embryo means it is formed by the fusion of male gamete with female gamete. Such zygotic proembryo means the embryo which is not well developed or which is young in condition. So such zygotic proembryo divides into many parts and each part later on develops into an embryo. Now we will see the advantages of polyembryony. Due to polyembryony, genetically similar plants are produced. With the help of polyembryony, the chance of survival of different plant species increases. Polyembryony is also used in horticulture for growing fruits. So in this chapter, we have studied different topics regarding the reproduction in lower and higher plants. So we have completed this chapter.